Hello and welcome back. This is our fourth video on photosynthesis. In the previous video for 6.3, we learned about the light-dependent reaction in photosynthesis. So in this video, we'll go on to the next part where we learn 6.4, the light-independent reaction of photosynthesis, also sometimes called as the Kelvin cycle. As usual, before we go into the content, let's have a look at the learning outcomes. I hope that by the end of this video, you will be able to explain the Kelvin cycle involving carbon fixation, reduction, and also the regeneration of RUBP. It's going to involve a few more steps, but I hope that you'll be able to follow along with the lesson and its contents. If you're ready, let's get started. So let's have a talk about what is a light-independent reaction. Sometimes called the Kelvin cycle, it is defined as the second of two major stages in photosynthesis following the light reactions, which involves fixation of atmospheric carbon dioxide and the reduction of the fixed carbon into glucose, which is carbohydrate. So it's saying it's in this part of photosynthesis, sometimes called Kelvin cycle, sometimes called C3 pathway, and it's going to involve fixation of atmospheric carbon dioxide. So kita akan ambil carbon dioxide, kita akan masukkan dia ke dalam sesuatu. And it's also going to reduce that fixed carbon into glucose. So daripada carbon dioxide yang kita ambil tu akan ada reduction dan kita akan pecah-pecah-pecah, susun sampailah dia menjadi glucose molecule. Nama lain dia adalah C3 pathway. Kenapa kita kadang-kadang gelar dia sebagai C3? Is because C3 refers to the first product of Kelvin cycle that is a 3-carbon compound, which is 3-phosphoglycerate. Also, sometimes you can refer to it as glycerate 3-phosphate. Nama dia C3 sebab ada carbon compound yang mempunyai 3 molekul carbon yang akan terhasil dalam proses Kelvin cycle nanti. This light-independent reaction will occur in the stroma of chloroplast of C3 plants during daylight. It occurs during the day, but light energy is not required for this part of the reaction. Example of C3 plants that we talk about here is paddy plants, wheat plants, or soybean plants. So once again, it is this part of the reaction in photosynthesis. C3 plants involve a lot of the plants that we already know about. Okay, so a lot of the examples of plants that you can think of are most likely going to be C3 plants. So one thing I would like to interject here, although this does not use any light energy, older books would call it the dark reaction. Kalau kamu pergi tengok buku-buku atau sumber-sumber yang lama, kadang-kadang dia akan gelar light-dependent reaction sebagai light reaction dan light-independent reaction sebagai dark reaction. But that is actually an outdated form of referring to them. Okay, so kalau kamu guna light and dark reaction, itu actually cara lama untuk merujuk kepada benda ini kita sekarang tidak terima sudah. Sekarang kalau kamu mau rujuk kepada kiri dan kanan ini, sila gelar dia sebagai light-dependent reaction and light-independent reaction. Kalau kamu tidak mau cakap light-independent reaction, sila gelar dia sebagai Kelvin cycle ataupun C3 pathway. If you don't need a light for light-independent reaction, what do you need? For light-independent reaction, we are going to require carbon dioxide. This carbon dioxide will be used to reduce to organic molecule via carbon fixation. We're going to need NADPH. This will be used as a reducing agent. We are going to need ATP. ATP will be the source of energy, and the product of light-independent reaction will be glyceroldehyde 3-phosphate, also known as G3P. So it will be a 3-carbon molecule. 2 molecules of G3P can synthesize 1 molecule of glucose. Just a very quick overall view before we go into step by step. For a net synthesis of 1 G3P, you're going to need 3 times Kelvin cycle and requires 3 molecules of carbon dioxide for carbon fixation, if you want to produce one glucose molecule, it's a 6-carbon molecule, you're going to need 2 molecules of G3P. To get 2 molecules of G3P, you need 6 times Kelvin cycle. And that means you require 6 molecules of carbon dioxide for carbon fixation. The importance of this is it will convert carbon dioxide to complex organic compounds, such as carbohydrates, by using chemical energy of ATP and NADPH. Saya bagi peringatan sekali lagi, light dependent reaction adalah untuk menjana tenaga, manakala light independent reaction adalah untuk menyimpan tenaga yang dijana. So earlier we already generated the energy in the form of ATP and NADPH. So now we're taking the energy from NADPH and ATP, and we're going to put it into the glucose molecules. So that's why the importance here is talking about putting it into complex organic compounds by using chemical energy. Alright, so the phases of Kelvin cycle. Kelvin cycle can be divided into 3 phases. The first phase is carbon fixation, the second phase is reduction, and the third phase is the regeneration of ribulose bisphosphate, or RUBP. Don't worry, we're going to look at each of these phases one by one. And for each phase, we actually can draw the molecules. So I would ask you, once again, unfortunately, to please grab a piece of paper, blank A4 paper preferably, and draw this diagram as we go along. So kita akan tengok setiap step dalam Kelvin cycle ni, dan saya akan cuba untuk explain dalam video ni. But if you have any problems, any part you don't quite understand, please feel free to ask in your own time, okay? So we'll go through this step by step, phase by phase. We'll start with the first phase of carbon fixation. Take a good look at the diagram before we start with the points. So kamu tengok di sini, kita akan start dengan carbon dioxide, ada RUBP, dan kita akan hasilkan benda ini. So, we start by saying 1 molecule of carbon dioxide will combine with a 5-carbon acceptor molecule, which is ribulose 1,5-bisphosphate, or we can shorten this name to ribulose bisphosphate, RUBP. Secara ringkasnya, carbon dioxide, 1 molekul, gabung dengan 1 RUBP. Dia akan hasilkan 6-carbon intermediate compound. It's going to create a 6-carbon intermediate compound. But intermediate means it's not going to last very long. It immediately splits and forms 2 molecules of 3-phosphoglycerate, which is a 3-carbon compound. So, bila ini masuk kepada benda yang 5 carbon ini, dia akan membentuk sesuatu yang 6 carbon, tetapi sementara sahaja. Benda yang 6 karbon itu terus terputus dan menjadi 3-phosphoglycerate. Hence, fixing of 3 molecules of carbon dioxide molecules will use 3 molecules of RUBP and would yield 6 molecules of 3-phosphoglycerate. Kita akan lakukan step ini 3 kali. 1 molekul carbon dioxide dengan 1 molekul RUBP, hasilkan 2 molekul 3-phosphoglycerate. Ulang step ini 2 kali, so that we use 3 molecules of carbon dioxide for 3 molecules of RUBP, and we have 6 molecules of this thing, 3-phosphoglycerate. That is what is meant in this segment. This reaction will have an enzyme to help it. The enzyme here is this very special enzyme called RUBP carboxylase oxygenase. Also have a nickname Rubisco. Sila ingat Rubisco ini ya. Dia adalah satu enzyme yang agak penting untuk kamu ingat. Okay? So after phase 1, we will have 6 molecules of 3-phosphoglycerate. Then we go into phase 2. Phase 2 is reduction. we are going to split phase 2 into 2 steps. Okay, di sini adalah step pertama dan step kedua. Phase 2 reduction, step 1 will be, we're going to take all 6 molecules of 3-phosphoglycerate. All 6 molecules of 3-phosphoglycerate will receive an additional phosphate group from ATP. So ATP bagi satu phosphate group, 3-phosphoglycerate akan menjadi 1,3-bisphosphoglycerate. So now we have 6 molecules of 1,3-bisphosphoglycerate. The phosphate group comes from hydrolysis of 6 molecules of ATP. And that brings us to step number 2. Step number 2 in reduction. 6 molecules of NADPH will be oxidized to form 6 NADP+. So sekarang elektron daripada sini akan masuk. The 6 phosphate groups will be removed from the 6 molecules of 1,3-bisphosphoglycerate. So phosphate keluar, elektron masuk. As a result, 6 molecules of 1,3-bisphosphoglycerate are reduced, and they become 6 molecules of glyceroldehyde 3-phosphate. So daripada ini menjadi G3P, which is something you've heard of before. So it's going to become 6 molecules of glyceroldehyde 3-phosphate. Overall, total number of G3P produced is 6 molecules. Only 1 molecule of G3P will be used by the plant cell to synthesize glucose molecule. Satu sahaja daripada 6 ini akan digunakan untuk menghasilkan glukos. Yang lain akan pergi kepada fasa ketiga. The other 5 must be recycled to regenerate the 3 molecules of RUBP in phase 3. The importance of this phase, apa yang dimaksudkan oleh tajuk di sini adalah kepentingan phase 2 of Calvin cycle, kepentingan the reduction part of Calvin cycle. The NADP plus and ADP that is formed in this process are returned to the thylakoids to regenerate NADPH and ATP in the light reactions. Fasa kedua ini penting supaya kita dapat balik NADP plus dan ADP. Kalau kita dapat balik NADP plus dan ADP, kita boleh teruskan untuk light reaction untuk menjana lebih banyak tenaga. Hence, the use of this phase of Calvin cycle is to supply more NADP plus and ADP for the light-dependent reaction. Okay, itu saja selingan. And then sekarang kita balik kepada Calvin cycle. Kita cakap pasal fasa ketiga, the regeneration of RUBP. Phase 3 is also known as regeneration of the carbon dioxide acceptor, which is RUBP. And it is done through a complex series of enzymatic reactions. Banyak step di sini dan banyak enzyme yang terlibat. The gist of it is that 5 molecules of G3P are rearranged to generate 3 molecules of RUBP. Daripada 5 molekul G3P tadi akan melalui penyusunan semula sampailah dia membentuk 3 molekul RUBP. And this step itself will require 3 molecules of ATP. At the end of phase 3, RUBP will be ready to enter the cycle once again. This is another diagram showing the Calvin cycle. Kita start dengan carbon fixation, followed by phase 2, which is reduction. This is when we're going to use the ATP and NADPH and produce 6 molecules of G3P. From 6 molecules of G3P, 5 will be reused in phase 3. 1 will be used later to form complex sugar such as glucose. Earlier, those were the steps in Calvin cycle. In this slide, we'll be talking about what we produce from the Calvin cycle. For the net synthesis of 1 molecule of G3P, we need to do the Calvin cycle 3 times. And for the 3 times of Calvin cycle, we need to use 3 molecules of carbon dioxide, 9 molecules of ATP and 6 molecules of NADPH. G3P will be produced by Calvin cycle, and G3P will be the starting material for metabolic pathway to produce carbohydrate, amino acid, and lipids. So, secara ringkasnya kita boleh cakap G3P ini yang terhasil daripada 3 round Calvin cycle boleh digunakan untuk membuat carbohydrate, contohnya glukos, tapi kita boleh juga gunakan G3P ini untuk membina amino acid dan juga lipid. Okay, so that is the net synthesis of 1G3P. So to produce one molecule of glucose, which is a 6-carbon molecule, you will need 2 molecules of G3P. Hence, you will need to do Calvin cycle 6 times. To do Calvin cycle 6 times, you need to use 6 molecules of carbon dioxide, 18 molecules of ATP, 12 molecules of NADPH, and all that to do 2 molecules of G3P, which will later be used to synthesize 1 molecule of glucose. So, ini untuk bantu kamu dapat gambaran lah untuk berapa kali kita perlu buat Calvin cycle untuk buat 1 molekul glucose ataupun berapa molekul G3P. When I was learning about the Calvin cycle for the first time, I find it very, very confusing because I don't understand why we need to do 3 cycle or 6 cycle. For that, I ask you to please watch this supplementary video on Nature's Smallest Factory, the Calvin cycle, through the TED-Ed channel on YouTube. This video, if you follow the link below, it will also be in the description, or if you follow the QR code, this video with its animation will be able to explain the Calvin cycle much better than I can. So please, in your time, watch this video, and I hope it will be able to help you understand why we don't just do 1 cycle or 2 cycle, why it has to be 3 cycles of the Calvin cycle to produce 1 molecule of G3P. So yes, please go ahead and do that, and I will move on to the next segment of the lesson. So when it comes to photosynthesis, light-dependent reaction and light-independent reaction is great. We can take the ATP and put it into our sugar molecules, but there are some problems when using the C3 pathway, and there are problems involving C3 plants. One major problem with terrestrial C3 plants is dehydration. So during hot and dry climates, to prevent dehydration, C3 plants will close their stoma. They'll close the stoma to conserve water. So when the stoma is closed, the concentration of carbon dioxide in the leaves will drop. You know, because you use the carbon dioxide for photosynthesis, and there is no new carbon dioxide coming in from outside because you close the stoma on the leaves. So concentration of carbon dioxide gas in the leaf will drop as carbon dioxide is consumed in the Calvin cycle. Carbon dioxide from atmosphere cannot enter the leaves, and so at the same time, because you are also doing the photosynthesis, the concentration of oxygen will increase. Sekarang banyak oxygen dalam daun kita because oxygen is produced by the light-dependent reaction. And oxygen cannot exit into the atmosphere from the leaves because the leaves have closed the stoma. So now we have the oxygen trapped inside the leaf cells, which becomes a problem. Why is this a problem? It's because when oxygen levels rise, photorespiration will occur in the leaves. This is a bad thing. Photorespiration occurs when the oxygen level is high and the carbon dioxide level is low. This has to do with that special enzyme in Calvin cycle, rubisco. So if you go back to the notes, phase 1, carbon fixation, enzyme yang gabungkan carbon dioxide dengan RUBP adalah rubisco. Masalah dengan rubisco Rubisco tends to bind with oxygen because Rubisco has a high affinity to oxygen. Kalau bandingkan antara oksigen dengan carbon dioxide, Rubisco lebih suka oksigen. Dia pandai curang. So, oxygen will act as a competitive inhibitor. Dia macam perempuan ketiga yang mengacau perkahwinan antara carbon dioxide dengan Rubisco. Dia adalah competitive inhibitor to this enzyme. So here, when your oxygen level is high and your carbon dioxide level is low, the oxygen will bind to Rubisco and cause the breaking of RUBP, which is a 5-carbon molecule, into one molecule of phosphoglycolate and one molecule of 3-phosphoglycerate, a total of 5 carbons in these two compounds. Sepatutnya, yang ideal, Rubisco akan pecahkan, Rubisco akan gabungkan carbon dioxide dengan RUBP. RUBP akan terus pecah kepada 2 molekul 3-phosphoglycerate. Tapi disebabkan oksigen datang dan kacau daun, dia akan hasilkan 1 molekul 3-phosphoglycerate dan 1 molekul phosphoglycolate, which is this 2-carbon thing. So that's a problem. That's a problem because phosphoglycolate then goes through a series of reactions using oxygen and ATP, releasing carbon dioxide. So, phosphoglycolate ni dia akan buat hal dia sendiri lah. Dia gunakan oxygen, dia akan gunakan ATP, dia akan keluarkan carbon dioxide. And it has been called photorespiration because it's going to occur in the presence of light. Dia berlaku sebab ada cahaya. And it will use oxygen and release carbon dioxide. So, kalau kita tengok bilangan carbon dioxide dengan oksigen saja, seakan-akan benda ini melakukan aerobic respiration. Tapi dia tidak menjana tenaga, malah dia membazir tenaga. The formation of phosphoglycolate caused the use of more ATP and even caused the release of the carbon dioxide with no formation of number of production of organic molecules, such as glucose. So it will reduce the efficiency of photosynthesis, or you can say it reduces photosynthetic output. And photorespiration can actually reduce photosynthetic potential yield by 30 to 40%. So kalau tiada photorespiration, 100% fotosintesis akan menghasilkan banyak glukos. Tapi kalau ada photorespiration, sebanyak 30 hingga 40% daripada glukos itu tidak akan terhasil dan tenaga akan terbazir. So, long story short, in C3 plants, sometimes photorespiration happens. Photorespiration happens, it's a problem because now there is less ATP in the plant and it produces more carbon dioxide but no glucose is being produced. Bazir bahan, bazir massa, bazir tenaga, tiada hasil. So that is a main problem of C3 plants. So this is a diagram showing the photorespiration in C3 plants. It's just a flow chart. Rubisco transfer oxygen to RUBP, resulting in 5-carbon molecule, which splits into 3-molecule phosphoglycolate. This will stay in the Calvin cycle like a normal thing. And it will also become 2-carbon molecule called glycolate. So glycolate will leave the chloroplast and goes to peroxisome. In the metabolic pathway, it begins in the peroxisome and then is completed in the mitochondria. Glycolate is broken down into water, and carbon dioxide. Unlike normal respiration, this process of photorespiration produces no ATP. So photorespiration will decrease photosynthetic output. Secara ringkasnya, slide ini jelaskan sekali lagi apa yang berlaku dalam photorespiration. Perlu hafal? As long as you know that it produces this molecule and that molecule and it's bad. Okay, that is the main point of photorespiration. C3 plants, like I've mentioned before, include these things. Okay, these are common examples of C3 plants such as wheat, gandum, soybeans, kacang soya, potato plants, and rice. Staple food for us here. Okay, so that is quite a long lesson and a lot for you to digest. Just a very quick summary of what we did in this video is we learned about the Calvin cycle. So make sure you go through the steps in the Calvin cycle and try to understand. Remember to watch the video that I linked to. And do remember that for us to do Calvin cycle to produce one molecule of G3P, we need to do 3 cycles of Calvin cycle, 3 molecules of carbon dioxide, 9 molecules of ATP, and 6 molecules of NADPH. And also remember, for C3 plants, Calvin cycle becomes a problem because sometimes we can have photorespiration when the weather is hot, there is too much oxygen and too little carbon dioxide inside the leaves. So I hope that is a good lesson for you. Take your time to understand the contents of this subtopic, come up with your own notes, and after you're done trying to digest the information, complete the tutorial question for Chapter 6, Part B, question number 2. That's all for me. Thank you very much for your time and your attention, and I will see you again in the next lesson. Happy learning, and bye!